again. So it's it's Tuesday night, and I'm down here in my little temporary studio making this recording, and the 8 o'clock howl has just finished. So I usually put off making this recording till after 8 o'clock howl. So let's see, where are we now? We finished up on Monday, and with what I represented on Monday as being the most amazing discovery of the 20th century, and I stick by that. And where we are, to summarize where we were, it's 1953, and discovered that DNA, which is a polymer of four nucleic acids, which we call A, T, C, and G, is this double helix. And it's the structure of that double helix, the way these two helices lock together, that makes DNA stable. And this molecule now we understand is the one that transmits genetic information. And so a cell's DNA is its blueprint. It's everything there is to know about that cell or organism or virus, whatever it happens to be. It's all contained within those nucleic acids. Now let's recall how we got to this most amazing discovery of the 20th century. For thousands of years, disease was thought to be caused by bad air, by miasma. Remember, Snow in 1854 provided evidence that cholera is transmitted by a living organism, by a living cell, and not by bad air. It took 40 years before Robert Koch saw this bacteria with a microscope. And those are the microscopes that were invented in the 17th century. And he was able to isolate this bacteria, culture it, that is to grow it, and this was the beginning of the germ theory of disease. And so finally, human beings had an ability now to manipulate, to control diseases by just simply filtering the water they drank. So over the next 15 years, however, there's increasing evidence of something which is, I'll call, semi-living, and which was given the name virus. And remember, it was discovered because of a disease on tobacco plants. And it turns out that the virus was so important to our story of how we came to understand things that are going on here, because it has only one property. It can reproduce. That's all viruses do. They don't think, they don't even consume. They just reproduce, and they use other cells to do that reproduction. So it's the simplest possible thing that we can study that will give us information about life. But the problem was it couldn't be seen. So with improvements to microscopy, so microscopes got better, optical microscopes got better, and eventually in the 1930s we got electron microscopes so we could begin to see things. And we had other ways to image things, which began in 1910, 1912, 1913, that area of time there, where we developed X-ray diffraction, which provided us the means to see viruses. And now the impetus, that's the important part, with those tools, the investigator said, oh, now I can maybe find out what this virus is. So that started all these things together. Now, along comes the development of organic chemistry, which with new separation technologies, which included ultracentrifugation, which we've talked about very briefly, electrophoresis, the ability to label molecules with radioactive isotopes. All of these things came together to play out so we could begin to understand not only viruses, but how viruses were put together. And what we learned were viruses were just a very simple assembly of proteins and nucleic acids. So the question became, what is the structure within the virus that gives rise to this ability to reproduce? And of course, the original bias was it must be the protein. But by the late 1940s, we'd come to understand, with all these tremendous techniques we had, we came to understand that it was the nucleic acids, that it was DNA, the nucleic acids that were responsible for the ability of something to reproduce. So that brought us to where we were last time, with the big question now with everything is always, this is what causes it, now what is its structure? Because properties derive from structure. So Crick and Watson concluded and provided just compelling evidence, along with stuff they got from other people, that the structure of DNA was a double helix. And now the sequence, this is the amazing thing. The sequence of nucleic acids that A, T, G, and C, the sequence on this necklace of DNA, these beads of C, T, G, and A, that is what codes for various amino acids which are put together to make proteins, and it's the proteins now which are the stuff that we're made of, that make us what we are. So this is 1953. Now this brilliant man, James Crick, taking the information that he had in 1953, laid out what's now called the central dogma of molecular biology. So what Crick did was he knows that within a cell, and they could tell this by optical microscopy, that in a cell, the DNA is localized. In certain cells, it's in the nucleus of the cell. In others, it's simply localized in the cell. In certain bacteria, it's simply localized in the cell. And that is not where protein synthesis goes on. So the DNA is separated from the area where protein synthesis goes on. So this brilliant man, Crick, concludes the following. The DNA, there must be an enzyme, a protein, that reads the DNA and does what we now call transcribe it. It transcribes it into some sort of a messenger molecule that is basically the message for a single protein. So it moves along this DNA strand, which codes for all the proteins that make up the body or the cell or what the organism, codes for all of those. And this enzyme moves along that and says, I need just this protein. And so now it codes for that little sequence of that. So it transcribes that DNA and turns it into a message. Now that message now is a much smaller molecule than the whole DNA sequence. So these smaller molecules now are somehow moved, transported, end up at the site of protein synthesis. And there must be another molecule that reads these messages, another protein, another enzyme that reads these messages and turns them into proteins. That is, it translates them. So the central dogma is DNA must be transcribed into a message, and that message must then be read and translated into a protein. That's the central dogma. It's the one we know about today. Now, Crick speculated that a DNA words are 3 letters long. Now, why did he do this? Well, there are 4 nucleic acids and 22 amino acids, 20 naturally occurring amino acids, but 2 more that occur on other places. So there are, if a word were 2 nucleic acids long, that would code for 16 possible amino acids, which isn't enough. On the other hand, if it was 4 long, it would code for 256 amino acids, which seems like a ridiculous waste, that we've got a lot of words that are either redundant or not necessary at all. So Crick said it must be 3. 3 is the right number. In Red Riding Hood's text, 3 would be just right. So 3 would code for 64 different possible combinations, and that seemed to be just right. Now, so here's this central dogma. Now, in the late 1950s, ribosomes were discovered. They were discovered by basically electron microscopy. And these are the very large enzymes responsible for reading and translating the message into proteins. So those were discovered in the late 1950s. Then in 1960, RNA polymerase was discovered. Now, that's the enzyme that's responsible for transcribing DNA. So And the way this turned out to work is that this RNA polymerase moves along the DNA and produces a complementary copy, but not of DNA, but of RNA. They're very similar molecules, but it reads and makes this RNA, and that RNA serves as the messenger that is now moved to the ribosome and is now translated and turned into protein. So now the skeleton of the central dogma is all laid out by the early 1960s. There it is. We know the machinery, we know everything about this. One thing left to do, that is find the 3-letter nucleic acid codes for the amino acids. So we basically have to break the codes. So in 1961, Marshall Nirenberg broke the nucleic acid codes, or he provided the mechanism to do it. He understood how to do it, showed others how to do it, and by 1966, the code was fully broken. So how did he do this? Well, here's the cool part that just blows me away. If I take a bacteria, and the one that is the common laboratory bacteria is E. coli. The E. coli, that's what we hear about all the time that causes, people eat lettuce that has E. coli on it, we have an outbreak and that kind of thing. But so E. coli is the one that people use. If I take E. coli and I just isolate, I just burst its membrane open, I take everything out of it, I just take the contents out of a whole bunch of E. coli, so there's no individual thing left, and I just pour them into a test tube, they'll continue during their biological function. That is, if I give them RNA and the ribosomes from the E. coli are there, they'll just go away and make protein. So this is so cool. We could just take the stuff and put it in there and off it goes. What Marshall Nirenberg did was he teamed with a synthetic chemist, and his name was Heinrich Matthey. And what Heinrich did was he produced 20 different solutions of amino acids. And these solutions contained only one radioactive amino acid in each one. So of the 20 possible amino acids, they were all in each solution, but each solution contained one radioactive one. All right. Now, what they did is they synthesized a synthetic RNA. And what they synthesized was what's called poly-U. And U stands for uracil, and that's just the equivalent of thymine, T, of DNA. So they synthesized this, and they have this sequence, which is just U, 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 or if it were DNA, it would be T, 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 T. Doesn't matter. Now, they take this RNA, and they throw it into each one of these test tubes. Well, the ribosomes in those test tubes say, I don't care that I'm not in a cell anymore. I'm just going to go along and I'm going to make protein. And it starts making protein. Now, after it's made protein, I've got these long strands of protein, and I can separate those from everything else. So these guys go about and they separate the protein from everything else, and they've got 20 batches of protein. And then they go along and they say, which one of these is radioactive? They go along, and here's the protein that's radioactive. Well, it turns out that that protein that's radioactive corresponds to the one that had radioactive phenylalanine in it. And phenylalanine is just one of the 20 amino acids. So they go, oh, UUU or TTT codes for phenylalanine. Well, the whole process now is straightforward. It's just a matter of synthesis of these various messenger RNAs, and some of them can be quite complicated to synthesize, so they have to synthesize these RNAs, put them in here, see which ones produce proteins, whether those are radioactive or not. So the whole process now is very straightforward, and people are doing this across the globe. But in no time at all, what happens is that by 1966, they've broken all 64 codes. So they know, and these are called codons. So every 3 nucleic acid codon codes for a certain amino acid, and obviously they're synonyms. So what we can get now is we get that there are multiple different 3-letter combinations that code for the same amino acid. So here we go. So it's 112 years, it's 1966, 112 years after Snow suggested that germs cause disease. And we've broken the code of life. Everything there is to know about an organism, cell, or virus is contained in the words written from its nucleic acids. And we can isolate these now. We can isolate this DNA. So there they are. On that DNA are the words. It tells us everything there is to know about that virus, that bacteria, ourselves. It's all there. So the last part of our story now is, we need to learn how to read these words. We've got the DNA there. We know what the code is. So now we can go along and we can read it. Now, when you learn to read, you started out reading very small books. And this is the same thing that's going to happen for us, is that starting in 1966, 1970, we began reading the DNA. And we began reading very small sections of DNA. But by the 1990s, this will culminate with us reading the biggest book, the one most important to us, which is called The Human Genome, where we began to read the human genome. So this is the 1990s, and that program ended about the time you were born. So that's going to be the last couple of lectures, is how we got to there, and now how can we take that information and turn it immediately into an understanding of the coronavirus? Because what we're going to discover is, because of all this work we did ahead of time, we were right on top of the coronavirus. We could read its DNA, or its RNA actually, but we could read it and we could tell things about it. So the last part of our story is going to be that, and then we're going to spend just a little bit of time looking at the future. So what's the next step? So we'll see you guys. Have a good E-Days, and we'll see you in the lecture in a few minutes, and then after that, we'll see you on Monday for house calls.